Now for our next panel. This will be the research panel. We have some experts up here to talk about the latest research. And I'm very pleased to introduce Ben Shaberman, who I've known for many years, from the Foundation Fighting Blindness. He will moderate this panel. Ben is the Vice President of Science Communications at the Foundation, a terrific advocate and communicator of all the science information to those of us in the lay audience. So, taking the baton.
Thank you, Martha. Give you a hug here. Thanks, Martha, for all you do for our community, the Carroll Center, and all you do everywhere. So, a theme that's come out today thus far, one of the themes is community. And I'm very proud to be part of a community that's really focused on advancing research to develop treatments and cures for Stargardt disease and a variety of other inherited retinal diseases. We bring together patients and families to help drive research around the world. We are a global leader in advancing that research. We currently fund about 90 projects around the world at prominent institutions. And excitingly, I remember when I started at the foundation many years ago, there was very little research that had moved into clinical trials. And today we have somewhere in the neighborhood of 50 clinical trials underway for promising treatments and cures, and many of those are targeting Stargardt disease. And one of the privileges I have is working with a really stellar group of researchers who are doing the painstaking work to advance these treatments from laboratories into clinical trials. And the three researchers we have with us today have all been, at one time or another, funded by the Foundation Fighting Blindness, so I'm very proud of that. And we're going to cover a couple of things. We're going to learn about what Stargardt disease is. We're going to cover some of the models underway, which are critical to advancing therapies, and then we're going to hear about some of the promising emerging therapies. But to start, I just want to have a brief introduction with each of our panelists. And to start off, directly to my left in this really beautiful pink dress with eyes on it, for those of you that can't see, maybe you'll tell us about the dress, Abby, is Dr. Abby Jensen. She's a PhD and an associate professor at UMass Amherst. So, Abby, briefly tell us about what you do at UMass Amherst.
So I'm a professor, so I teach neurobiology courses, so I'm a neurobiologist. And my lab has been studying retinal development in zebrafish for a number of years, and we've developed several models of retinal degeneration diseases. Most importantly for this group, Stargardt's disease. And I'll tell you a little bit about that in a bit.
Great. Thanks, Dr. Jensen. And next up is Dr. Jason Commander. He's an MD, PhD, and a clinical researcher at Mass Eye and Ear.
Yes. Thank you for having me. So I'm a clinician scientist at Mass Eye and Ear, and I specialize in inherited retinal diseases. I see patients, I participate in the conduct of clinical trials, and I have a research laboratory for diagnostics and therapeutics for inherited retinal diseases like Stargardt's.
Great. And then at the end of our row of panelists here is Dr. Peter Zhao. He's a vitreoretinal surgeon and assistant professor of ophthalmology at Tufts Medical Center. Peter?
Thanks for having me, Ben. So I'm the youngling in the panel here. I came to Boston about 1.5 years ago, and prior to that, I was in training, residency, and fellowship at the University of Michigan. And so for any alumni out there, go blue. It's been a great year. So I did a fellowship in inherited retinal degenerations and then a second fellowship in vitreoretinal surgery. And I'm a professor at Tufts. And I have clinic, 80% of the time, 4 days a week. I teach residents and fellows, medical and surgical retina, and then I'm in research 1 day a week. And my research focuses on two things. One is trying to develop better functional vision outcomes, so we can have better tests of functional vision in patients with inherited retinal degenerations. And then number two is I partner with Nadia Waheed. We have a collaboration with MIT and Jim Fujimoto where we have prototype OCT devices. These are devices that image the retina at 2.7 micron resolution, and we're looking in patients with IRDs and specifically Stargardt disease, looking whether we can identify new biomarkers with that higher resolution and try to understand the pathophysiology.
Thanks, Peter. And to start off, so we can help people understand different ways that we may approach Stargardt disease therapeutically, can you tell us what Stargardt disease is, how it impacts the retina?
Sure. So Stargardt disease has had, as some of the other panelists have alluded to, it's had a couple of different names over the years. Stargardt disease, Stargardt macular dystrophy, juvenile macular degeneration. And they're now all thought to be within the same condition, and they're all caused by pathogenic variants or mutations in a gene called ABCA4. And the gene ABCA4, it provides the instructions, it codes for the protein. And this protein participates in the visual cycle. And the visual cycle, if you think about an engine cycle, it's something that's constantly running. A car engine, it turns gasoline into energy. And the visual cycle does something very unique. It turns light into electrical signals. And so the light that enters our eye goes through this visual cycle and it turns into neuronal signals that eventually produce images on the brain. And when this protein ABCA4 is not functioning properly, not functioning at 100%, some of the products from the visual cycle end up stuck. They get stuck in the photoreceptors and they don't complete the cycle to be recycled. And then those products and their toxic byproducts accumulate, and that leads to retinal degeneration. And so there's the gene side of things, it's just ABCA4, but then what is classically been thought to be Stargardt disease, we've actually learned the ABCA4 gene, when it's not working, it can produce several different appearances of the retina as well.
And just a follow-up question. Vitamin A is such a crucial nutrient for vision. We all need vitamin A for our retinas to work properly, but ultimately vitamin A is sort of the culprit or byproducts from it, or the culprit in Stargardt disease. Can you say a little more about vitamin A's role?
Yeah, of course. So vitamin A is needed in the visual cycle. So vitamin A is, it becomes it gets eventually turned into a molecule called retinal, not to be confused with retina. But this molecule, retinal, is in a special form called the cis form, and that gets transmitted over to the photoreceptors. The light hits it, and in a synchronized reaction, the cis form is turned into a trans form. And then that trans form needs to go back and be recycled back into the cis form. So the vitamin A is necessary for vision. And there are patients who have vitamin A deficiency and they start to lose vision because they don't have vitamin A. But at the same time, the ongoing nature of the cycle and the fact that there are byproducts that are toxic in Stargardt disease, that may play a role in the degeneration.
Right. So yeah, we need vitamin A to see, but the byproducts from it are are toxic, especially in people with Stargardt disease. So would you say that not supplementing with vitamin A is the right course of action for people with Stargardt disease?
I agree. So I would say, you don't want to supplement extra beta carotene or extra vitamin A. So if you're taking any vitamins or supplements over the counter, look on the label and make sure there isn't extra beta carotene or vitamin A in there. But I wouldn't go so far as to say, don't eat your carrots, don't eat your spinach. I think that's going a little bit too far.
Right, right. Good advice. Good advice. Thank you. And so, Abby, you're involved in creating a model of Stargardt disease. And I think sometimes when we think about models of disease, it sounds kind of technical and maybe not too exciting. But they're really critical to advancing therapies into humans. And can you tell us a little more about models and the work you're doing?
So having animal models of disease really allows us to carefully identify mechanisms of disease. In order to develop therapies, you need to know those mechanisms that are underlying the disease process. So there have not been very good models of Stargardt's disease. The go-to animal model for research is mice, rodents. These are nocturnal animal, and they're rod photoreceptor dominated. So those photoreceptors you use for nighttime vision. Stargardt's is probably, I think we'd all agree, is mostly a cone, at least initially, of disease. And so because rodents have almost no cones, it has not been a very useful model for understanding cone dysfunction. So a number of years ago, we had made a model for retinitis pigmentosa. We disrupted a gene. We made a model for Leber congenital amaurosis, another disease. But it came to me that maybe we studied zebrafish. And you might think, zebrafish? Why would somebody study it in a fish, right? Zebrafish have a cone-dominant retina. They use the same vitamin A is required for their vision. The retina is designed in the same way as it is in us. We thought, well, maybe zebrafish would provide a great model for Stargardt's disease, a cone-based disease. So I still have those other models, but we haven't been studying them much. I still turn over the lines all the time, but now we study Stargardt's disease, and we knocked out the ABCA4 gene using molecular genetics. And we can really see the cells very well, the cone photoreceptors, and from an early age, follow what's going wrong with them. We're about to publish our paper, at least submit it. A remarkable finding that we've seen is that the sensory portion of these cells, where the vitamin A process is going on and the photons of light are captured in this compartment, is longer than normal. Now, this is opposite to almost every retinal degeneration disease that you will see, where this compartment shortens in rods or cones. So in Stargardt's, we're finding that this compartment is longer than normal. We think we understand why that's the case. So I think this might be a very powerful model for understanding the cellular basis of dysfunction.
So once you create a model in zebrafish or you understand it better, do you think you can use that model to test therapies as well at some point?
I think there are a couple of ways that this can be a useful model. One is we can control the environment of these fish, right? We can control the light levels that these animals are experiencing, the wavelengths that these animals are experiencing. So I think for the first time, we can identify whether or how light can affect the progression of the disease. Obviously, as a person having no light is not useful. The other one is we could test small molecules using this model to see how they affect these cone photoreceptor cells. So that's the other one. So those are the first most powerful uses, I think, for this model.
Sure, and what you learn in these models and testing these therapies can help inform other therapy developers that are working to advance therapies ultimately into human trials, I would think.
I think so. We found this, and I won't say why, it was kind of a fluke. We have this observation that even normal cone photoreceptors, this outer segment region that normally captures the photons of light, normally presents a eat me signal that you would never expect to be on a live, viable cell. But it's there in normal cells. And that pattern is disrupted in Stargardts. But the significance of that eat me signal being there all the time has broader implications for other retinal degeneration diseases as well. If cells enter that space, that shouldn't normally, but like in age-related macular degeneration, encounter an eat me signal, those cells will start eating those cells, the outer segments of those cells, which is not a good thing, right? Normally they're protected from that process. So these were unexpected findings.
And I think that's an important part of science. You set out with a goal and a hypothesis, and then you learn new unexpected things along the way. Well, thank you for talking about your zebrafish models and the work you're doing there. Jason, we give you the all-important topic of emerging therapies, and there are a lot of different ways that therapy developers are targeting Stargardt disease, small molecules, gene therapies, even cell therapies. Can you talk about what's out there, what's moving forward?
Yeah, absolutely. And just to pick up on a point that you started, when I started as a doctor, we would say, my template in the electronic medical record was, we discussed research about Stargardt disease. And then I had to change it. We discussed research about Stargardt disease and planned clinical trials. And now it says, and we discussed research about Stargardt disease and ongoing clinical trials. And so it's a really whole different world out there. There's so many people working for you all, and I'm excited about it. Past, present, and future. So these are some highlights, not totally comprehensive, but some past trials that I'd like to highlight is there was one to take the broken gene and put it back into the retina using a cargo vehicle called a lentivirus. And I'm going to come back to those genetic-based therapies at the end, and that one wasn't successful. Another one, a kind of group of trials, was for visual cycle inhibitors. And these are things that are chemicals, they're pills that are meant to slow down the chemistry of vision that Dr. Zhao was talking about. And a couple of them have been tried, and they didn't reach their endpoints for slowing the disease. There was another trial for a complement inhibitor, so that's a particular inflammation pathway. That one, it's now known as Izervay, got approved for dry, age-related macular degeneration, not Stargardt disease. But they did try it in some Stargardt's folks, and I didn't hear how that went, but that's usually not a good sign, I haven't heard any update on that. So as far as taking a step forward to trials that are ongoing but fully enrolled, like meaning they've gotten all of their people, there is another visual cyclomodulator that Belite Bio is testing called Tinlarebant. And this decreases the retinol transport to the retina. The trial's called Dragon. And I think they have all their people, and they're supposed to have data this year. Hopefully it's different or better than the other visual cyclomodulators that didn't make it. Nanoscope is a different company pursuing a strategy called optogenetics. And it's a weird term, but optogenetics is a strategy where you put artificial light sensors in the remaining parts of the retina. So say all your cones are gone, there's nowhere to put the ABCA4 gene anymore. Let's put artificial light sensors in the inner retina that remains. And they've treated 6 patients with their MCO10 to the bipolar cells, which is like the next level of cells. and I don't know the results for that one. Now, there's a set of trials that are enrolling now. One is Alkeus. So they have an oral pill, a molecule called gildaretinol. It's a modified form of vitamin A that is supposed to be able to complete the visual cycle but not produce the toxic byproducts that can lead to the health problems in the retina. And they have 4 clinical trials. One of them is done, and they have results for that, and they're trying to get FDA approval based on those results. Their second trial has finished, and they're expecting more results from that next year. And there's 2 more trials, one of which is an open label extension study, meaning that people can join the trial, but it's not randomized. All the people are getting the drug. That's not a placebo versus drug kind of trial. And that one is going on at a number of sites in this country. So Ocugen is another company. They have a potential therapy based on the RORA gene, R-O-R-A. And this is the modifier gene approach, where they think that if you put this gene back into the retina, it doesn't fix the original problem, but may have a positive influence on the progression of the disease. It's thought to help cells that have a defect in lipid metabolism or oxidative stress or inflammation. And that one is called the Guardian, and it's enrolling in Arizona and Texas. There's also a trial at the National Eye Institute in Maryland for metformin, which is a diabetes drug. And that one, at least according to clinicaltrials.gov, which is a good resource, is currently enrolling in Maryland and Michigan. Another good resource, while it's on my mind, is the clinical trials webpage at the Foundation Fighting Blindness website, which Ben Shaberman maintains. And I look at that all the time. It's a great place to look, and it's very reputable. You're going to get good information there. So now the future. So I'll admit that I'm partial to the genetic therapies, to the gene-based therapies. So the original problem in Stargardt disease is that there's mistakes in the DNA that codes for this ABCA4 gene, and both copies of it are broken in Stargardt disease. And so an elegant way to try to treat this problem is to put a copy of the missing gene back in. And that was what was successful for Luxturna, for the one approved gene therapy for a different gene. The acronym for that one is RPE65. And so the folks that were working on this about 5-10 years ago said, well, why don't we do the same thing for Stargardt disease? Let's put a correct copy of the ABCA4 gene back into the retina. There's only one problem. The DNA sequence for the ABCA4 gene is really long. It's over 6,000 DNA letters long. And the cargo capacity of the virus that was used for Luxturna is only 4,000 and something. And so, put your scientist hat on for a minute. What would you do?
Find something capable of delivering larger.
Yes, that was exactly what was done. They picked a different cargo carrier that could hold more, and that was the lentivirus trial that Sanofi did. And that wasn't successful. So now the field is going back to the original cargo carrier, the adeno-associated virus, AAV. And there are 3 different companies that are taking kind of creative approaches at it. The first one is Splice Bio. And they're going to, and these are trials that are potentially starting this year or next year, perhaps some of them in Boston. So Splice Bio is using what's called a split inting technique, where you put the 2 pieces in, and then the 2 pieces, when they're inside the cell, they recombine to form one protein. And the key question is, does enough of it find its partner and get together to make a difference in the disease? And so that's what's going to be tested. Ascidian, another company, instead of having the split inting technology where the protein comes together, they're using a trans-splicing technique where 2 pieces of the RNA come together. So, at a different stage of the production of this protein, you put in a special RNA in it containing the first half of the gene that's correctly spelled, and there's a special bit on the end of it that makes it latch on to the back half that's already in the cell. and it comes together and it produces the full correct protein. And then the last one along those lines is Avant-Garde Bio. And they have a different dual vector approach where they're putting the two halves in there. So those are cool. Astellas Regenerative Medicine, they may be doing a stem cell trial. I haven't really heard about that. And if you do look at clinicaltrials.gov, there is a pair of clinical trials for the folks in Florida peddling the stem cells that are not reputable, and so don't go for that. That's my list.
Well, that's a pretty long list. And yeah, just to emphasize those sort of bogus opportunities in Florida, if you ever hear of a clinical trial where they want you to pay for the therapy, stay away from it.
Well said.
But it sounds like there are a lot of promising genetic therapies that are coming down the pike, and hopefully in the next year or two we'll have clinical trials underway for those. But I want to ask you a basic question, Jason, about people who are interested in participating in clinical trials, because you are an investigator in several trials for a lot of different retinal diseases. What advice would you give to somebody who's thinking about a trial?
Well, I would take a step back and say that the clinical trials is just one piece of the whole set of homework that you have. And the other speakers today highlighted really well the technology, the resilience skills, and all the things that you can do. And when I have a patient who doesn't qualify for any trials, I say, come back in 2 years, take care of yourself until we find something better for you. And so, so that's a big part of it. And then you get into your personality too. So depending on the stage of what you have, you may not even qualify for the trial. You may even be too mild or too severe, depending on the approach, the particular approach. But then there's also risk calculations too. So these are tests of potential therapies that we don't know if they work yet or not. And and some people, they want to be on the leading edge of things, and some people want to wait and know whether it works before they get involved with it. And that's totally cool too. If you do want to get involved, then just, ask your eye doctor or ask a specialist. Stargardt is rare, so depending who you're actually talking to, they may not know. Go to clinicaltrials.gov. If you find something interesting, contact the person on the email there and ask them. They may want you to provide a copy of your eye exam or that kind of thing to see if you might qualify. But yeah, that's the advice that I give.
Right, and I think an important point that you're implying is clinical trials are research. And so when you enroll in a trial, you're part of the research. Hopefully it'll work, hopefully it'll help, but it is research. So we're still learning about the therapy when you're in the clinical trial, and people need to keep that in mind. I think another important point that you made is that a lot of doctors, especially a private practice retinal specialist, for example, may not be that familiar with the clinical trial process. And it's really important to get connected to perhaps a clinical researcher or somebody at an academic research center like you or Dr. Zhao, who really understands the therapy landscape and what it's like to be in a clinical trial. And the Foundation Fighting Blindness is a great resource for that. If you happen to be in the Boston area, you have two of the best in the city here, so they're great resources too if you're thinking about getting involved in a clinical trial.
Yeah, I agree with you, Ben. I think one of the most important things for people to do is to get genetic testing, because there are some other genetic retinal diseases and potentially other categories of retinal diseases that can look like Stargardt disease, but they're not Stargardt disease. And so it's important, number one, to see an inherited retinal degeneration specialist who has seen many of these patients and many of these cases, and to get the genetic testing through them. They're the ones who's going to be able to put together what they're seeing in your eye together with the genetic testing result, which is getting more and more complicated as we have these panels that are testing 350 plus genes. but be able to put together that information and say, do you have Stargardt disease, or might you have something that looks like it but isn't quite it? And clinical trials are, especially in the gene age, I'll call it now, of gene editing, they want to make sure you have, ABC and a variant in ABCA4, because they want to make sure that the treatment that they're proposing, the treatment that they're testing, is targeted to the right patient.
Right. Thanks for emphasizing the importance of genetic testing, and the Foundation Fighting Blindness for people in the U.S. has a no-cost genetic testing program if you're interested. I am thinking it is about time to take questions from the live audience and or the chat. And I see some hands going up, several in our live audience.
So I'm gonna take one question from the chat first, because we take one from the live audience for the first session. So there's a really good one. How can we find doctors who have experience with Stargardts? Are there any compiled lists anywhere?
That's a great question, and I'll take the first stab at that. On the Foundation Fighting Blindness website, fightingblindness.org, under the resources section, there is a list of retinal doctors around the U.S. that are familiar with inherited retinal diseases, and most of those should have some familiarity with Stargardt disease. And you can always just reach out to us directly by sending an email to info@fightingblindness.org for whatever your question might be about a retinal disease. But that's a great way to get some doctor suggestions as well. I don't know if anybody on the panel has more to say about finding a retinal doctor.
I think that's going to be your go-to list. If the doctor or if there aren't any doctors in your area in the United States, I think probably your best bet would be your local academic center, if there isn't anyone on that list that is near you.
Right.
Yeah, I'll come over.
Okay.
Thank you very much. It's been extremely informative. You've talked about the gene therapy, which holds promise for individuals with inherited retinal diseases. Some of the research and trials have actually led to the restoration of vision. And I think for all of us, particularly parents and those with Stargardts, that's the holy grail, right, of the research and trials. As you mentioned, a lot of the published research indicated that the obstacle for Stargardt's patients in particular is delivering the large amount of genetic material in order to make a difference. You spoke about some of the progress that's being made, but I was wondering if you could address for us the potential promise of CRISPR gene editing.
Yeah, I can take that one. I've worked on that. So it's probably more efficient if you can get a correct copy of the gene back in to the retina just by adding something from the outside. That might be a preferred method. But if the gene is way too big to deliver into the retina, then you get into the gene editing category where you have to fix, or you try to fix, the DNA that's already there. And there was a really landmark trial called the Brilliance trial where that was done for a different disease, a different gene. CEP290 was the name of that one. And what was presented at conferences is now is being submitted as a paper. Is there's some signs that some of the patients were helped, and we think that there was activity in of editing the gene in the eye. And there's a couple generations of the CRISPR technology too. It's gone from CRISPR to base editing to prime editing, and there are various companies working on various aspects of those. One of the tricks of the genome editing is that they tend to be more specific to particular mutations, like you fix one part at a time. So one trial might target a subset of people. For the prime editing, they're trying to figure out how to do bigger and bigger pieces all at once. So you may get a whole group of people. But if you can deliver the whole thing, then you could hopefully apply that to everyone. And it's a matter of what's going to be more efficient and safer in the end. I don't know of any CRISPR trials planned in humans yet, but there's certainly lots of people are doing things in the laboratory with it for ABCA4 and other macular dystrophies. Yeah.
More questions?
Hi, thank you. Hi, first off, thank you all for all the work you guys are doing. I don't have Stargardt, my son does, but for all of us that have been working on and living with it for years, we rely on you guys. You guys are doing an amazing amount of work, so thank you for all your effort, first and foremost. My question is, and I don't know whether it was Jack or Chloe or someone in their story, talked about the feeling you get after you go to the doctor for the 10th time and you're putting through all those tests, and is it really worth going? So my question for you, and on behalf of my son, is what is the right number of times we should be going to the doctor? Is the benefit there the data pool that you guys can get from what we're doing by the time we visit every 2 years, if we should do that? Or is it every 5 or 10? So I don't know if you guys have a recommendation. That's what I'm looking for.
That's a really good question. I think to speak to a general point, it's really important for patients to stay engaged as much as possible. The example that I always think of is at the start of the COVID pandemic. I looked. Everyone's at home. No one's doing anything outside of the house, and I think on the New York Times website there was a running list of the clinical trials for the COVID vaccine, and there were 22 trials going on within, I think, a month of the pandemic starting. And when I saw that, I was very optimistic that at least one of them would come through, and that's through the engagement of the entire scientific community of people who are willing to try a vaccine that had never been tried before. And I think it takes engagement from researchers dedicating their careers to this. It takes engagement from patients, and it takes many foundations working on this together and bringing industry and everyone together to come up with a treatment or a cure. I think from a clinical standpoint, it's reasonable to follow every 1 to 2 years. What do you think, Jason?
Yeah, I agree, and probably even more important than the frequency is the approach of the eye doctor when you're in the room with the kid and the parent. And the doctor that started the department at Mass Eye and Ear 50 years ago, he taught me something very important that maybe there's some eye doctors watching this, I hope there are, and he taught me, show them what they have, not what they've lost. Because as doctors, you go and this is wrong, and this is atrophy, and this is degraded, and this is missing. But I always start out with the big fluorescence picture of the retina, and I say, look, over 95% of your retina is working fine. You're going to keep your side vision. For you, your reading vision's the issue. And we're going to track that over the years. And it changes, but it tends to change really slow, so I don't even see people for every 2 years. Sometimes when people are having a hard time, if they're in the more denial stage or kind of getting used to it, sometimes maybe I'll say 1 to 2 years, hoping they'll come back and get more engaged. But I think if you make it positive, then whether it's 1 year or 2 years or whatever, then hopefully it's helping them. And you're the parents, so you get to decide what's good for them. And when I do 2 years, I usually say see your local doctor in the off year. Just, a yearly, some kind of minimal check would be good too.
And just a follow-on comment about engagement. I think we have to remember these diseases, Stargardt disease is rare. And the more data, the natural history information we can gather, the better equipped we are to launch clinical trials. And seeing doctors like Dr. Commander, Dr. Zhao are important. You can also, if I can give a plug for our patient registry, myretinatracker.org, if you can put your information about your disease in the registry, it's a no-cost, highly secure registry, that will help you get that really valuable information out to the research community so they can better understand the condition. So that's a good way to engage, and obviously you can do that from the comfort of your own home and computer. More questions?
Yeah, I do want to read a question from the online chat. It says, is there any research as to why some patients present later in life with symptoms?
Great question.
Yeah, I think there's quite a bit of variability, as we've heard from our patient panelists. Some patients get it when they're children, some people in young adulthood, and some in late adulthood. And I've even seen some family members, where they have a grandson or a granddaughter with, Stargardt disease and symptoms where, they came in for an unrelated reason and they have, a very, very mild form of Stargardt that maybe they didn't even know about. And so the big question out there is, why do some people get, very mild disease and some people get it, when they're children and get more severe disease? Some of it is the environment, and I think some of it is how the ABCA4 gene and how the retina deals with, those toxic byproducts. I think even between two siblings who have it, it can be very different. And then I think the other part of it is what part of the ABCA4 gene is affected and how is it affected. Some of the variants that we see, or mutations, they can be milder because the function of the protein is reduced only a little bit, whereas other variants, the function of the protein can be reduced more severely.
Yeah, I totally agree with that. There was a paper by one of the experts in this, Rando Alec Metz, where he tries to lay out which mutations are more severe and which are more mild. And I don't really recommend that patients go try to figure out where they are in that scale. Really, you want to look at what you have. And one of the best things is the fundus autofluorescence picture, where you can see if there's any black parts where it's not healthy at all versus other parts. And a retina specialist will tell you where you are as far as mild or severe.
Yeah, and I'll add another comment about the foundation. We are funding Dr. Alec Metz to better map those mutations to potential severity of disease.
Great.
More questions? We have about a minute and a half left.
Just a quick question. My son, who has Stargardt's, is seeing a retinal specialist in Toronto and did get genetic testing there. His doctor suggested that his parents each get tested for our genetics. Do you see a value to that?
Yeah, so it depends a little bit on the specific situation, but so Stargardt disease is a recessive disease, so you're supposed to inherit one broken copy from each parent. Sometimes you could get it faked out where you could have two mutations that come from one parent and 0 that come from another, so you may be looking in the wrong place if that's what happened in your particular family. Usually that's called segregation testing, and so you test one parent, you see, oh, there's one from one parent. If you want to really go all out, you can do the other parent and see there's one from the other parent. But that's just proving that you inherited it from both parents and that it's not a fake out. So usually it's okay without doing that, but if you really want to know, or if the disease doesn't look like Stargardt disease, but the genetic testing is saying it is Stargardt disease, that might be the reason why, that it's not a real diagnosis, and that can help untangle it. Yeah.
So thank you, Jason. One more question and we'll wrap up. And the enthusiastic person.
That's me. I'm very enthusiastic. So I have a question. Everybody who has Stargardt's, and I'm the mother of one of the panelists, has an experience with an ophthalmologist. I can remember the PD ophthalmologist accusing Jack of lying, which was really a pivotal moment for me in my parenting. The ugly mother came out. My question is, I also work at the Carroll Center, and I think the retinologist that ultimately diagnosed Jack, he wrote Stargardt's on a piece of paper, gave it to my husband, and said, you should go home and research this. My question is there any way for the foundation to have, you've got the American Pediatrics Association, right? Is there any way for the foundation to educate and inform optometrists, ophthalmologists, and others about inherited retinal diseases and places to go if you get diagnosed, so that the parent just doesn't get handed a name on a thing on a piece of paper?
Well, thank you for that question. And I'm happy to say that about 5 years ago, we launched a department. It's called the Professional Outreach Department, and I'm part of that, actually, or at least was. And that's exactly what we're doing. We're educating eye care professionals at all levels around the country about inherited retinal diseases and all the great resources that are out there, whether it's genetic testing or the Foundation's chapter network and all the clinical trials. So yeah, it's a big challenge. There are tens of thousands of eye care professionals out there, but we do have a resource dedicated to that. So thank you. And I think that will conclude our research and science session. Abby, Jason, Peter, awesome discussion. Thank you. And thanks to all of you out in the community for attending and your interest in eradicating these difficult macular conditions.
Thank you so much. Really interesting information. Maybe 15 years ago when I first started fundraising for the foundation, there really was almost nothing in the way of clinical trials. There's so many going on today. We don't know. All of us want treatments like yesterday. We don't know when they're going to get into the clinic, but I do know one thing. If we do not support research, they'll never get there. So I am very passionate about supporting research, even if it's not going to help me. I certainly want it to help others not have to go through what we go through.